Hello friends, welcome to BMH Learning. In this video, we will talk about estimation of total sugars by Eynon and Eynon method. Sugars are inherently present in different food products. Glucose and fructose in the fruits represent reducing sugars, while sucrose represents the non-reducing sugar. They are determined or estimated by Eynon and Eynon method, which measures sugar as reducing sugar and total sugar as invert sugar. Principle: invert sugar reduces the copper present in Falling's solution to cuprous oxide, which is red and insoluble. The sugar content in a food sample is estimated by determining the volume of the unknown sugar solution required to completely reduce a known volume of Falling's solution. Glucose and other sugars are capable of reducing oxidizing agents and thus are referred as reducing sugars, and this property is used for the estimation of sugars. The cupric ions in Falling's solution are reduced to cuprous state, which gets precipitated as red cuprous oxide. Only reducing sugars reduce the copper solution. The method is suitable for estimation of sugars in fruit and fruit products. Reagents required and their preparation. Falling's solution A. Dissolve 69.28 gram copper sulfate in water. Dilute and make the volume to 1 liter and filter. Falling's solution B. Dissolve 346 gram of Rochelle salt and 100 gram sodium hydroxide in water. Make volume to 1 liter. 1% methylene blue indicator. Dissolve 1 gram in distilled water. Make the volume up to 100 ml. 45% neutral lead acetate solution. 225 gram of neutral lead acetate is dissolved in distilled water. Make up to 500 ml. The chemical is used as clarifying agent. Potassium oxalate solution. 110 gram of potassium oxalate is dissolved in distilled water. Make the volume to 500 ml by adding distilled water. It's used for neutralizing excess of lead acetate. Standard invert sugar. 10 gram of sucrose is dissolved in 100 ml water. For hydrolysis, add 5 ml concentrated HCl. Allow to stand for 3 days at 20 to 25 degrees centigrade. Make the volume to 1 liter. Standardization of standard invert sugar. Pipette out 25 ml standard invert solution into a 100 ml volumetric flask. Into this flask, 50 ml distilled water is added. Also add few drops of phenolphthalein indicator. Neutralize the solution with 20% sodium hydroxide until solution turns into pink color. After this, acidify it with 1 normal HCl by adding it dropwise until the pink color disappears. Make up to 100 ml by adding distilled water. The concentration of invert sugar we are getting here is 2.5 mg per ml. Standardization of Falling's solution. 5 ml Falling A solution is mixed with 5 ml Falling B solution in a 25 ml conical flask. Into this flask, 25 to 50 ml water is added. Then we heat the flask. After this, standard invert sugar solution from the burette is added dropwise into the flask till the solution turns brick red. Then few drops of methylene blue indicator are added. Continue adding invert solution dropwise. As soon as the blue color disappears, Note down the titer value of invert solution. Repeat the titration and calculate factor for failing solution. Procedure for estimation of reducing sugars. 10 to 25 gram of food sample, which can be fresh fruit or a fruit product, is taken. Grind it in a pestle and mortar or blend in a blender. After this, add 100 ml distilled water. This solution is then neutralized with 1 normal sodium hydroxide. Then boil the solution for 1 hour with stirring. Replace the water that is lost during evaporation. Then cool it and transfer to a 500 ml flask. Make the volume up to 500 ml by adding distilled water. Filter the solution through Waterman filter paper. Now, once all this is done, pipette out 100 ml liquid from filtrate into 250 ml flask. Into this, 2 ml 45% lead acetate is added. Let the solution stand for around 10 minutes. Then, few drops of potassium oxalate solution are added. Make the volume to 250 ml with water and again filter the solution. Again 2 ml 45% neutral lead acetate solution is added into solution filtered in previous step. Then into the same 250 ml flask add 5 ml each of phthalate A and B. Also add few ml of water. Flask containing mixed phthalate solution is heated on hot plate. The sample solution is added drop-wise from the burette until the faintest blue color remains. Then 2 to 3 drops of methylene blue indicator are added. Titration is continued till the color changes to brick red. At the end point, note the readings and calculate the reducing sugars. This was all. Thanks for watching.